Hello everyone, welcome to the DBMCI. Today in this session, I'll be discussing about the PCR techniques because the COVID pandemic is out there and it is bothering us since almost more than 1 year now. And whatever is out there, we all know, I don't know how to say it, but we should have an understanding of how the RT PCR that is done for the COVID testing is working. So in today's discussion topic, first of all, I'll tell you the basic about the PCR, what PCR is, that is a polymerase chain reaction, and then how PCR is related to RT PCR, which is done for this viral genome, because RT PCR is usually done for the RNAs, and viruses have the RNAs as a genetic material. And then we will just be discussing about the positive and negatives of the RT PCR, how CT value is related, and what are the current hot discussions that are going on regarding to the CT values. So welcome to the session. Myself, Dr. Varsha Gupta, and let's start our journey for the SARS-CoV-2 and the real-time RT PCR as well as the CT value. So first of all, what is PCR? You can say the origin of RT PCR came from the PCR. PCR stands from the polymerase chain reaction. As the name is suggesting, there is this polymerase, right, like the DNA polymerase, RNA polymerase, you all know, and there is a chain reaction that is going on repetitive over a period of time, and that we calculate over the graph that gives us the PCR. It is one of the simply and most widely used technique nowadays. It detects as RNA as well as the DNA sequences. PCR is used for the DNA samples, but there is another extension of PCR, that is RT PCR, that uses RNA as the sample. But if the question is asking whether we can use PCR for RNA, the answer is no. RT PCR is for RNA. Okay. It requires few hours, approximately 3 to 4 hours are required to get the results from the PCR sample, as compared to the earlier blotting techniques, which usually used to take 2 hours or so. It is very highly sensitive method. Even if you start up with very tiny amount of the starting material, it can amplify the starting material and give you positive results. Right? It is used majorly nowadays for the paternity testing, detection of the mutated oncogenesis, bacterial and viral infection, identify pathogens and the parasites such as Trypanosoma cruzi, that is the causative agents of Chagas disease, and meningitis, that is the causative agent of the meningitis, right? And it is used for comparing the samples in the forensic cases, such as the rape cases, murder cases, where you get the semen samples, blood samples, and they are in very minute quantity. It is also used for the HIV testing. So for every test to run, we need to have some basic necessities, the requirements we call them. The first requirement is the DNA polymerase, because we are working with the DNA, we have to amplify the DNA. So DNA polymerase, and specifically for PCR, we need the thermostable DNA polymerase. Taq polymerase is the kind which we use in the lab. Magnesium, DNTPs, because as we know how the transcription and the replication process begins, you have to add the bases to it. For those you need the DNTPs. Primer is used. DNA template we needed to, which is the actual first and foremost requirement. Template is the sample. Template is your sample, and the thermocycler, also known as the PCR machine, is needed. Procedure: initially you do a prep in which the sampled DNA is added to the test tube along with 2 primers, right? And then you add a thermostable DNA polymerase, or Taq DNA polymerase is added to the sample test tube. And then the extension of primer sequence occurs in the presence of by adding DNTPs in the thermocycler. right? And the thermocycler has a set of cycles, right? The first mixture is heated, that is a heating cycle, to almost as say 92 degrees Celsius. That is the denaturation of the double-stranded DNA. Because double-stranded DNA we cannot replicate, so we need to first of all denature the double-stranded to make it the single-stranded, and that single-stranded is used as a template, right? And then mixture is allowed to cool very slowly, and that is not done to for the re-annealing of the DNA, but for the annealing of the primers that are used to elongate. And these primers, for our case when we are talking about the COVID, is specifically for the COVID sequences, because in a sample you have your RNA sample that is coming from the SARS-CoV-2 virus, as well as you have another RNA samples present in the sample that you have collected from the oropharyngeal or from the nasopharyngeal swab. So you just don't have the SARS-CoV-2 virus RNA, but there are another things to it. So specific primers are used because now we know the entire genomic sequence of the SARS-CoV-2 virus, and we take certain genes, 3 genes. I will show you the map, which according to those genes we have made the primers, the forward primer and the reverse primer, and also a probe. So the primers are specific for the COVID virus, and then this above cycle is repeated and over and over. So actually there are 3 steps. First is the denaturation, in which double-stranded is denatured and we make it single-stranded one. Then the annealing of the primers is done. Then elongation of primers are done by RNA polymerase, which result in the copies of the original DNA strand. All these 3 steps make one cycle. And when you do the PCR amplification, you almost do 30 to 40 cycles of this to reach a certain threshold value that is already pre-defined by the scientist. That threshold value is above the baseline. The baseline is the one when you don't have the sample DNA but rest everything, and that threshold value is below the quantity of the amplified DNA, so that you don't have too much of the background noise in the sample. So that threshold value is very, very important. Okay. So this is how the amplification of the PCR in the PCR cycle is done. So there is the cycle 1 and the cycle 2. So what you are doing in the cycle 1, you have the double-stranded DNA from 3' to 5' end, and the other one is running in the opposite direction because the strands, they run in the entire parallel direction. So heating is done. Heating is done to almost 95 degrees Celsius. Strands are separated. They are cooled down slowly. Primers are added, and that cooling down is approximately 75 degrees Celsius. Then a thermostable or heat stable DNA polymerase is added. Again, the temperature is 75 degrees Celsius, and the elongation is done for both the strands. So let's say we are talking about the strand 1, that is 3' to 5' direction. The primer is 5' to 3' direction. And when after the elongation, from one strand you have strand 1, and also this is the new strand. Same thing happens with the strand 2. You make one more strand. So you have strand 2 and then you have new strand. So from 2 strands of a single double-stranded DNA, you got 4 single strands, or you can say you got 2 double strands. And then you repeat the cycle again. So these 4 individual strands after denaturation becomes the elongation, and these 2 becomes, now these 4 individual strands becomes 8, and or you can say the 2 duplex becomes the 4 duplexes. 1, 2, 3, 4. So every time you're running a cycle, you are amplifying it times 2. Same thing if you repeat the cycle 3, then these 8 single strands become 16 single strands. So it is like 2 raised to power n the number of the cycles. So you can imagine if you run 35 cycles, right? So 1 step, 2 step, 3 step and you do it for 35 times, so then it is 2 raised to power 35, or you can say that is 34 billion DNAs we are talking about here. So this much amplification is done by the PCR. That's how we able to manage the startup DNA can be a very tiny amount because it could be amplified in the reaction mixture via the PCR cycler. It is a highly sensitive material, that's what I mean. Very minute quantity of the sample can be started as a sample and that can be amplified. And to do this reaction, actually a master mix is recommended because reaction is like you have a 96 well plate. So many reactions you can done at one time. And master mix is a mixture of you have your DNTPs, magnesium, ions, you have your DNA polymerase, RNA primer, all those things those are listed as the requirement. They all are there except your template DNA or the sample DNA. So first what you do is you in the 96 well plate, which I will show you, you add all the master mixes. 3 are kept as a control, and then let's say you have 10 samples. So you can run 10 samples at once by labeling which well contains which sample. Okay. The major challenge in running the PCR reaction is the GC rich region. Let's say your template strand has multiples of GC, then they tend to form this hairpin structure. Now, if there is a hairpin structure, then there is a difficulty of DNA polymerase to go and bind because now it is sort of a secondary structure. DNA polymerase cannot accommodate itself in and do the entire replication process. So, in order to destroy the GC-rich regions, usually for those sequences, there is this particular chemicals that are added to the reaction mixture. We add the DMSO, glycerol, and betaine. They destroy the secondary structure, then helps us to in the remediation process of the GC-rich region, and then we can run the PCR reaction. Okay. Now, another problem with the PCR is since you have this heat stable DNA polymerase, okay, and we know that the elongation should be done at, let's say, 72 degrees Celsius, 75 degrees Celsius. It depends, it varies from instrument to instrument, okay. So let's say 75 degrees Celsius, you want elongation to take place, or you will say the DNA polymerase should start elongated. But because DNA polymerase start working even before you want the elongation. So what it does, it first of all binds at the unspecified region of the template or the sample DNA. Then before even we need the reaction, we want the reaction to proceed further, it starts the replication process. So that decreases that creates a problem because you already have the products coming. And since we want reaction to start at the elongation phase only, so what is done by the scientists is they have added an antibodies to the DNA polymerase, and those antibodies make the DNA polymerase inactive. So unless these antibodies are attached to DNA polymerase, they are not going to go for the elongation. But as soon as, let's say, 75 degrees Celsius or the desirable temperature is reached, these antibodies they get detached from the RNA polymerase, making or setting the RNA polymerase free so that it can start the replication process. That would what I mean by the hot start PCR. So to increase the sensitivity or to get the specific amplification, this is done. That by adding antibodies, we are deactivating the DNA polymerase unless the desired temperature is reached. Now, that was the PCR. Then we did the hot PCR. Now, what is RT PCR? In the COVID case, our sample is not the DNA but the RNA. PCR works with the DNA only. So, what we know from the genetics is we can convert the RNA into DNA via the reverse transcription process using the enzyme reverse transcriptase. So, RT means reverse transcriptase. This is reverse transcriptase process, reverse transcription, polymerase chain reaction. When you combine those two, in first half you are converting, in RT you are converting the RNA into DNA, and that DNA is to be amplified via the similar mechanism that we have just discussed. So RNA is used as a template in RT PCR, and very important thing is the purity and the quality of RNA template. It's very important factor. How much the sample collection is of prime importance to ensure the reliability of the RT PCR. And that we will see that how come you have the false positive, how come you have the false negatives. More or less it is depending upon the purity of the chemical reagents. The technician who is running the RT PCR is well qualified and trained for the sample collection method was adequate. The right time was there for the sample collection. So all these factors, they contribute towards the results. Now there is one more term now, qPCR, or you can say, what is qPCR? qPCR is a quantification term, because when you are running the thermocycler, you are increasing the amount of the DNA. But how will you know how much DNA has been increased so far? So for that we add on a new technique to it, that is a quantification technique. We add the fluorescent labels to it. So more products are formed, so it will fluoresce more. That is the quantification. That is quantification PCR. The same thing, same benefit is harnessed by the RT PCR. So sometimes you call it as a QRT PCR or real-time RT PCR. Those are all the same names. Little bit differences there. RT PCR, the starting material is RNA. When we say the PCR, starting material is the DNA. Okay. Well, so there are two ways for the quantification. One is the dye-based, another is the probe-based. Okay. So dye-based is usually a green dye, which is not much used. The most better reliable method is the probe-based qPCR technique, in which the probe that is a hydrolysis probe, and it has a two-part to it. So this is, let's say this is a probe. Probe is a kind of a primer that has a particular base sequence to it. So it can be A, T, G, C, C, A, T, A, and these probes are specific for the SARS-CoV-2 virus, so that they can attach to the template DNA strand. Okay, and on one side you have the fluorescence, and then other side you have the quencher. So via the Frick technique, fluorescence, resonance, energy transfer, fluorescence is going to give you the fluorescence. It is going to fluoresce. But since there is a quencher attached to it, the quencher absorbs all the fluorescence given by the fluorescence probe on one side. So what in this RT-PCR does is, when the DNA polymerase is in the elongation phase, there is this probe which is attached to this DNA template strand. So you have here your DNA pol1, DNA polymerase. So it is going on, going and going and going and going. Boom! It strikes to the DNA. This hydrolysis probe. It is going to break this by the exonuclease activity. So the fluorescence portion and the quencher portion are just now floating in the solution. Now fluorescence portion is fluorescing, but there is no quencher next to it for the quenching. It's dead now because the entire probe is broken down now. So more the fluorescence, it means the amount of the DNA that is made, or you can say the product is higher. So fluorescence is now directly proportional to the amount of the DNA that is being made by the DNA polymerase enzyme, or you can say how many, with the increasing number of cycles, the fluorescence is going to go increases. Okay, it will increase. So for the COVID-19 testing, the samples from the nasopharyngeal swab or oropharyngeal swab and other ways, sample is being taken, and then the sample is taken actually to the testing center. In this is the diagrammatic representation of a sampling center, Okay, now this person, as you can see, has the headgear on. It has in the hand a ice box to it, and in this ice box there is a ziplock. That ziplock is containing a tube that contains the sample, the swab sample from the patient. So it is a three tier of the packaging is done. Then the sample is transferred into the lab via this one way openable pass box. In these actually the pharmaceutical factories or the testing laboratories, this is kind of a window which is called as a pass box. It can be a static pass box, it can be a dynamic pass box. This is a static pass box. It opens from one side and this person leaves this in the window. There is a little bit portion in the window, and the person inside the lab, now he is inside the lab, is now geared up with the elbow length gloves, the full body overalls, face shield with the respirators. Now it is going to open the one way pass box and collect the sample. The sample is matched with the person's identity so that the results are delivered to the right person. Okay. Then RNA isolation is done. That is again a process to know. And in next slide, in the upcoming slide, I will show you how the RNA sample is isolated from the patient sample. Because when we take the samples, if let's say from the nose it is taken, you have the cells, you have the mucus, you have lots of things actually along with the real SARS-CoV-2 virus sample. So isolation need to be done, and then the qualified and the validated instruments are used. So this is the tube that is containing the nasopharyngeal swab. Most of you have seen it, I'm pretty sure by now, how it is taken, and then it is broken and left. It contains a solution actually in the bottom. So in this hood, using the gloves and everything geared up, this person is trying to do the isolation of the RNA. This machine is showing, this is a thermocycler or PCR. After the RNA isolation is done, that is placed in the 96 well plate and the thermocycler is done. Okay. And this is a screenshot of the computer. And if you can see here, when I'm talking about the 96 well plate, this thing here is representing the 96 well plate. Little bit cut, 10, after 10 you have 11, you have 12. So these are used for the blank. These are the blanks to compare for. And 8 in this side and 8 in this side. In this you have samples from the different person. Okay. Very strong and quality driven system and processes for the data integrity and the traceability is done. Bio-waste are discarded as per approved guidelines, and the reporting is done as per the government requirements. Okay. And good news is the Simpla is announcing the launch of one RT PCR kit known as the Vera Gene. Earlier Simpla's kits, antibody detection kits and antigen test kits are there in the market, but the antibody detection kits and antigen test kits are not reliable for RT PCR. Antibody is definitely not used for the RT PCR detection. Antigen test kits, also known as rapid test kits, they are not really very reliable, and doctors really do not recommend it. RT PCR at today's time, if you are asking me, is the only way, or you can say the best available method out there with us to detect the SARS-CoV-2 infection. Okay. Now this slide is showing you how the RNA extraction is done from the sample that was collected from the patient. Okay. So in this mix, this is the sample, okay, from the nasopharyngeal swab, and lots of vortexing is done, and the incubation was done using a lysate buffer. That is making it the orangeish color. And what this lysate buffer contains is, it has first of all your phenol, guanidine, isothiocyanate, an RNase inhibitor. So it has 4 things. I'm going to write it down here for you. This has the phenol, guanidine, isothiocyanate, and RNase inhibitor. Why RNase inhibitor? Because we don't want our sample RNA to get degraded by the RNase enzyme if it comes in there. So inhibitor is added. Phenol, guanidine, isothiocyanate, or written here is, all these are going to lyse the every extra thing that we don't want other than the RNA. The proteins, it's going to degrade all those things, okay? And it is made set at the room temperature for some time, and then vortexing is done. Then if you can see here, then what is this? This is the spin column. This entire sample is poured over the spin column. What this spin column has? This spin column has this white bed, that is the silica bed. What it does is it just binds to the RNA, sample RNA. Rest everything comes here at the bottom. That is the lysate, and that is usually we discard it. Okay, then all the entire RNA are now stuck in this silica bed. Wash buffer is added to it so that anything that extra other than the RNA is still there in the spin column, washed out in the bottom, which is the discarded. Finally, the elution buffer. The elution buffer is added. This elution buffer allows this RNA, sample RNA, to come at the bottom, which is now your purified sample RNA that is used for the PCR amplification and the testing. So here, this is the 96 well plate, an example of the 96 well plate, not exactly. In this, first of all, the 3 rows, I have tell you, this matrix is like the 8:12. Okay, so 3 out of 12, 3 are used for the controls. So rest of others, like 8:8 matrix, is used for the sample. And now you can well imagine how many samples you can run at one time. So every well you add the master mix, and then you label it which sample are, how many, let's say 10 samples you have in the lab. So you can run all those samples together in this 96 well plate. So master mix contains your reverse transcriptase, that is first going to convert the RNA into DNA. Then the primers, forward primers and the reverse primers are going to get attached, right? And then also the Tachman probe, which has the fluorescence and the quencher to give you the fluorescence signal, that is the real-time PCR thing. And then DNA polymerase and DNTPs. DNA polymerase is going to use the DNTPs for the extension of the DNA. So RNA is converted into DNA by the reverse transcriptase. Then it is heated so that the double helix RNAs are not there, or in our case actually it will be single stranded DNA only. And then RNA polymerase is going to come and bind to the site recognition site. And then you have the reverse primers and the forward primers. They are going to attach to their places. Tachman probe is going to attach at its own place, and then the elongation step will occur in this PCR cycle. And this is the computer interface. Every lab, there are so many versions of this thermocycler or PCR instrument. So here is, in this instrument you keep this 96 well plate here, and this here little screen here is showing you how much cycle, and also it shows the graph that how much the fluorescence signals is being increasing. And usually we set the cycle to, let's say, maximum of 30 cycles we run for the PCR. Okay, so this is the genome there. This is the genome of the SARS-CoV-2. And whenever all the kits that are available in the market are based upon just 3 kind of genes, they are targeting 3 genes, the RDRP gene, E gene, and the N gene. And based upon those 3 genes, we have the forward primer, reverse primer, and the probe. So we have 3 sets. This set corresponds to the RDRP gene. This set is used to target E gene, and this set is for the N gene. So based upon which kit we are using, that kit is specific for either of these 3 genes, and that is written on the label of the kit. Okay, and now this is the real-time graph that is we see on the monitor. And if we read at the graph, there is this baseline in the blue that does not, this is the PCR, that's the control without the template. And above that, if you see the red line, that is the exponential curve which we get via the fluorescence signal. So this exponential phase, at this exponential phase, scientist has come up to a threshold value. Threshold value is above the baseline, but it is well below before you see lots of the background noises. That is like because too much of the cycle has been run, there is too much of the sample in the small Appendorf tube. So CT value is fixed, and for the SARS-CoV-2 virus it is 35. So anybody which have the less than 35 value is positive and above 35 value is negative. So what do we mean by that? So imagine, so initially you have these 2 patients. This patient sample, the RNA sample is, let's say, 5 nanogram. In this sample has the 50 nanogram. This is definitely more. So this will require less number of cycles to reach to threshold value. Am I right? So less number of cycles is required when the sample has more amount of the viral load. So CT is indirectly proportional to, or you can say is proportional to 1 over the starting amount of the target DNA. Meaning higher the CT value, it means the lower the viral load, lower the infectivity. Lower the CT value means the patient already had too much of the RNA in it. That's why it reached the CT value very soon. So it is inversely proportional. Okay. So CT value is what? CT is a threshold value, that is number of the cycles that are required for the fluorescence signals to exceed the specific threshold value. According to IEP report, the ICMR has advised CT value less than 35 may be considered as positive and over 35 is considered as negative. Right? So if we took at the worldwide, it is anywhere between 35 to 45, the cutoff value. Okay. And there is this published paper which is showing that the viral load in the sputum, it correlates with the risk of the COVID-19 progression. What they did is they took the samples from the mild, moderate infected patient and the severe critical patients, and they measured the CT value here, CT value on the y-axis, admission in hospital follow-up. So dark color is the mild and moderate. Mild and moderate, you will see the CT values are higher as compared to the severe patients. So it means lower the CT value, that is patient is more serious. And here the second graph is about the probability of disease progression. So when you have very low value of CT, there is a high probability of disease progression. When there is a low value of, high value of CT, then there is a low probability of the disease progression. Okay, so that is about how the CT value correlates with the COVID-19 progression. Okay, but how accurate are our results? The accuracy of results depends upon the analytical specificity and the sensitivity. These two things in RT PCR makes it very, very special and most of the times the results are accurate. Okay, so this is the table which will explain us how the false positive and the negatives are coming. So if the person is diseased and the test is positive, so definitely this is true positive result. Okay, the person is infected. When the person is not diseased and he goes for the RT PCR testing and the rest comes negative, so the person is true negative. Now the other two left columns are the gray areas. The person is not diseased, but the test comes out to be positive. So this is false positive. False positives are not very bad because the person is not diseased. It is not going to infect same person is not going to infect the other people around him or her. So it is okay. It is bearable. The problem is when the person is diseased and the test comes out to be negative, it means false negative. This is dangerous in this pandemic because there can be a person having the false negative report in the hand but actually he is a COVID patient, and when he will walk in the streets, you might be the one getting infection from the false negative patients. So these are the critical person and we have to take a serious action on this. False positives are why? Because of the technical errors, because of the reagent contamination. We do redo the test 1 day, 2nd day, 3rd day, right? Then we can really trace, okay, no, the patient is okay and this is a false positive result. Okay, but these false negatives are very critical and they happen majorly because of the type of the specimen used. Because initially earlier I have told you also that the RNA sample purity is of very great importance. That will determine the accuracy of the result. Okay, and the time of sample collection. And the false negatives usually come from in-sampling techniques. So laboratory technicians who are collecting the samples, they should be very well trained at how to collect the samples. The transport should be very accurate upon the storage conditions in which the sample has been stored and taken down to the facility unless they are ready for the testing. They should be handled and transported and stored very carefully so that samples doesn't get destroyed. Okay, and in RT PCR, it is true most of the time because the dual tests are done before the reporting. First, the screening is done and then again the testing is done for the confirmation. Okay, now this is a very recent paper in the January, just 4 months back, 2021. In this, the Shweta, Pooja, Namita, Davar, Tanu, they are saying there is no correlation between CT values and the severity of the disease mortality. So that is a big blow. How come the CT value is not related to the severity of the disease? They have run a test on the COVID-19 patients, right? They say that patients who died had significantly lower CT value. That is okay. Then patients who survived, but again they had significantly shorter duration of symptoms before testing. So we don't have to just consider the CT value. That is indicating we have to again go and check the symptoms also. Now, to clarify this, Dr. Prakshit Prayag, who is consultant of infectious diseases at Dinanath Hospital in Pune, is saying that CT value definitely does not correlate with the severity, but it correlates with the infectivity. How much infective that patient is for others. Okay, now this is the discussion from Michael Mina from the Harvard, and he gave some comments which were wrongly published and it created a chaos in the entire world, I'm telling you. Okay, but he was saying the same thing, that the CT should be published along with the positive results to better idea of the viral load. Okay, and he is saying that sampling error, sample storage conditions, and the severity of infection. All these variables are going to influence the CT values, and they should be discussed before declaring the patient as a COVID positive patient, right? And they say that the PCR kits that are available in the market, you cannot compare them. They always are not going to have the same threshold value. Okay. And now for the easiness, the RT PCR report should have a QR code, so they don't have to read it, just scan it. Tamil Nadu government cancels the permission to the private lab because the patients or people are getting the false negative reports and they are running down in the street. And the Odisha government facilitates online access of the COVID test results, is making it easier for the public. Now the thing is, we know that the symptoms of the allergies, cold, influenza, and COVID-19. This is like, you can say, is a painful thing that all these symptoms are the same. That makes it like very crucial for us to rightly diagnose it in the timely manner so that we can save the patient and also we can save the people who are living around that person. Okay, so we have to find a ways for this management and the prevention of the infections. How do we holistically manage it? We know there are two types of tests in the market. One is the direct test or the diagnostic test, that is RT PCR, or you can say the rapid antigen test kit that is available in the market. And second is the indirect test, which is the antibody test kit. Actually, that does not test you for the COVID infection or not. That is just measurement of the antibody level inside your blood. And that antibody might have been in against the COVID is there because either you were the patient before and you have recovered from the COVID infection, or you have gotten the vaccination that is creating the antibodies inside the body. Okay, so antibody test is the test to develop the antibodies, right? And the rapid antigen kit is to detect the specific viral testing, but both are not reliable for testing the SARS-CoV-2 infection. So in order to manage this disease and to prevent the infection, if you have the symptoms which are similar to the COVID, like the fever, cough, shortness of breath, vomiting, anything, right? So RT PCR is the test that we are going to go for the diagnosis, right? If you know about that you have developed the immunity in the past, you can do the antibody test, but that is not the diagnostic test. Okay, if you want to join the office or the college, go for the RT PCR test as well as go for the antibody test. Why both? RT PCR test will tell you that you are negative, you are not going to infect the others. Antibody will give you the assurance that you have a specific antibody level inside your body to fight for the infection if you come in contact with the infected person. So you do both. Before and after travel, both times you go for the RT PCR, because when you go before travel, so you know you are negative and then you are not spreading the infection in the society. After the travel, make sure that you never came in contact with an infected person and you are safe. So before and after travel, both times you do the RT PCR testing. So guys, thank you for listening. Be safe, be aware, and take care of your families. Every time I'm praying to God that everybody be safe and sound and the harmony comes back on the Mother Earth. Thank you. Bye-bye.